Hello everyone, welcome back. Today I'll be explaining chapter 5 of the fifth objective, enzyme. Now, what is an enzyme? Well, enzymes are very essential biological molecules. So they are essentially a protein. In essence, they are a protein. Enzymes help us to catalyze, to make metabolism faster in our body. So basically, any chemical reaction that take place in our body depends on enzyme. And if there were no enzymes in our body, then these chemical reactions would take more longer than usual, right? So enzymes act as catalysts. So we can describe enzymes as a biological catalyst right, that increases the rate of chemical reaction. And we have to note a very important factor is that enzymes are not changed by the reaction. In other words, they catalyze the reaction, but they do not get changed or deformed or become something entirely new all of a sudden. So the enzymes themselves do not change, and it's not change in the process of the reaction. And therefore, we can see that enzymes can be reused. Enzymes are constantly reused, right? You have to know that enzymes are proteins. Now, these proteins are involved. They are involved in all metabolic processes and reactions. Now, you might be wondering, what is metabolics mean? Well, matter really mean change. Molecs basically mean reaction, right? Change in reaction. Now, this change in reaction, this metabolism or metabolic reactions are basically, an example of metabolic reaction is respiration. Now, respiration takes place in all of our body cells and every type of different cells, because respiration is needed, right? Aerobic respiration is needed for oxygen, right? And with oxygen, this respiration needs the oxygen, and it's going to produce ATP as the molecule. Now, ATP stands for adenine triphosphate, right? So there's that. And adenine, ATP is an energy molecule. It is a universal energy carrier or molecule, you can say whatever you like, but it is an energy carrier molecule, and it's basically used as a form of currency or a form of money in the biological world, right? So enzymes require ATP, and they are not an active process. They use ATP to convert a substrate into a product. Now, there will be new terms that you have been familiar with. This is the substrate. Now, substrates are basically the reactants, or the molecules that you will be turning them into a product. Now, there are two types of catalytic activities by enzyme. Now, and there are two types of enzymes. Enzymes which actually break down large molecules into smaller ones or other molecules. This is called catabolism. And for enzymes which build up the molecules from small to large, small molecule build up large molecule. Now, this is called anabolism. So remember, in metabolism there are two types. If the enzyme break down from large to small, it is called catabolism. And if you build it up from small to large, it is called anabolism. And these are two things that you have to firstly understand. This is not in your syllabus, however, but you do have to understand that kata meaning break down, ana meaning to build up. Right. Okay. So now that you understand that enzymes are very, very essential molecules that are important for the metabolic process or metabolism reaction, and they function as biological catalysts, they increases the rate of reaction. Now, if enzymes were not here, then the reactions would take way longer than usual, and our bodies would not function as it is supposed to function. So it's not as efficient if we don't have enzymes. Now we have to know about the mechanism on how enzyme actually works. So let's start by saying how enzymes work or mechanisms. Let's look at the mechanism of how enzyme works. Now enzyme action, this is called enzyme action. Enzyme action is actually it works on the basis that the substrate, right, the substrate will bind to the active site of the enzyme, and then it's going to form an enzyme substrate complex. Now this basically means that the enzyme is actually holding the substrate molecules together by some various bonds between the enzyme and substrates. And then the next stage in this is that the enzyme will form a product complex. Product forms. So basically the product is not actually formed yet, but the product is formed, but it's still attached to the enzyme. Now this is called the enzyme product complex. Now enzyme substrate complex can also be written in short as ESCs, right? But it's better to use the long form if you write those in the exam. Now thirdly, finally, only then when the enzyme product complex is formed, only then the product will leave the active site, right, and the product is removed. Now, so obviously it's really hard to visualize if you don't have a picture. Now we have to actually look at how the enzyme works. So I'm going to draw for you the diagram. So let's say that this right here is our so let's say this right here is our enzyme, and then this, and then this, and this is our substrate, or basically the molecule that we are going to be breaking down, right? So you can see that there's, first you have to know where the active site on the enzyme is. Now this part, this region right here, let me highlight this for you. This region right here, this is called the active site. And this is where the substrate is going to bind to the enzyme. Now note that this whole diagram is basically drawn in 2D, while proteins, all the proteins and molecules are actually 3D in nature. So this is a very oversimplified diagram, but this helps you to understand it really well. Now firstly, the substrate will approach the active site of the enzyme. Now what happens is that, now secondly, remember the active substrate enzyme substrate complex is formed. Enzyme substrate complex is formed, so it's going to be attached. It's not going to be attached yet, right? You can see it's going to attach to the active site, and these lines that I'm drawing right now are actually bonds that are holding the substrate in place. So basically the substrate will go and join with the active site, where basically bind to the active site. Now, note that you have to use words, terminology like bind to the active site. So substrate binds to the active sites, and that's a very key word and key definition that Cambridge would want you to use. Now, so this is called the enzyme substrate. So the enzyme and substrate are actually bonded chemically together now. It's being held. Now, next thing is that the enzyme will work its action, and then it's actually going to become something like this. So the substrate molecules, we're breaking down 1 and 2. Now you can see that these are not linked together anymore. Do you see that there are no more links here? Here it was linked. Here the original molecule was linked together, right? But here we can see that it has separated into 1 and 2 pieces, into 2 different type of molecules. See that different products are formed. Now there are 2 products that are formed from basically 1 molecule, right? Now this is called the enzyme product complex. This is called the enzyme product complex because the products, however, when the products are formed, but they haven't left the active site of the enzyme. Now only then the bonds will be broken between the products and the enzyme. Only then will the substrates, the products will leave. So the products will leave from the active site. So products leave at active site. So basically this is the whole process that you have to understand. Now note that here you can see that in this highlight, okay, I'm going to use another color. When I highlighted this, you see that the curve and the enzyme active site, right? The substrate and the active site of the enzyme are very complementary to, they are basically the same. In other words, they are complementary to each other. This triangular curve is actually the same or similar to the active site of the enzyme. Now, this leads to a very important concept that you have to understand, is that this is called the enzyme specificity. Enzyme specificity is basically that not all enzymes are going to bind to different types of substrate. So one substrate will bind to one type of enzyme, but so many substrates cannot, to so many different types of substrate cannot bind to basically to essentially any enzymes. So what I'm trying to tell here is that if I draw another molecule, such as something like this, if I draw something like this, something of the sort, like this, you can see that the active site is actually not going to fit them, right? The active site is not going to fit. And this leads to a very important concept. Now this is called enzyme specificity. Enzyme specificity. Now enzyme specificity relies on the complementary shape. The complementary shape and the fit of the active site. So we can say that the substrate and the active site, or basically the enzyme, has to be complementary to each other. Now if they are not complementary to each other, then obviously they won't bind and the products will not be formed. Now this is a very important topic, so always refer to enzyme specificity when you talk about how or why enzymes do not bind to all types of molecules, and that each enzyme is specific to that one molecule. So for example, if glucose C6H12O6, the enzyme that is responsible for catalyzing this reaction will only catalyze enzyme that is responsible for this reaction, for respiration, will only work for respiration and will not work for other types of reaction. So there is specificity. So we can see that the enzyme is showing specificity. Sorry. Right. And now this is actually called the key and lock model. And always try to include this in your answers as well. In your level, you don't have to know this, but I'm just going to add a little bit extra, which is that you can see that when the substrate actually binds to the active site, we can see that the enzyme slightly changes shape. As it binds, as the substrate binds, right? As the substrate binds to the active site. Now this is called an induced fit model, but for your syllabus you don't have to know about this, but it's good to know for additional information. Yeah. So now we have to actually look at the effect of enzyme activity. The effect of enzyme activity. Now there are many different factors which affect enzyme activity, and one of them is actually temperature. Now how does temperature affect the enzyme? Well, if you increase the temperature, if temperature is increased, you know that the substrate molecule kinetic energy will increase. When kinetic energy increase, what does this mean by this? Well, what I'm trying to say is that when kinetic energy increases, the substrate move faster. And when temperature increase, it is not only the substrate which will have gained more kinetic energy, it is also the enzyme. So we can see that enzyme and substrate both will gain kinetic energy. And then this makes that the enzyme and substrate will move faster. If they move faster, this leads to a very important concept, is that if they move faster, there is a higher chance of the substrate and enzyme colliding with each other. And if they collide, reaction occurs. Now this is so-called the kinetic theory. Okay, so when collision occurs, right? When collision occurs, products are formed. So when temperature is increased, the frequency or the number of collision that occurs per second, right, of collision also increases, and therefore thus the product is formed more readily or more faster. It is formed more readily. Okay, now that's the first of the temperature. However, we have to look at the side effects or the downside or the cons, disadvantage of using too high temperature, a temperature that's too high. So let's say for our normal condition for our human body, our body cells, they work around the temperature, they work best around the temperature 30 to 37 degrees Celsius. Now if were the temperature to be even a bit more, let's say 45 degrees Celsius, 50 degrees Celsius, then when the temperatures become really, really high, it can actually change, it actually changes the active site. So basically the shape of the active site, right? It actually kind of, I'm gonna use an ice cream analogy here, because when you eat ice cream in the summer days, it's basically the sun heat, the radiation will melt off the ice cream, and so the ice cream will actually change its shape. So this is basically just like the enzyme. The enzyme will change its shape when the temperature is really too high, and it will not work anymore. if the active site changes shape, that means no more substrate can bind to it. No more substrate can bind because the active site was altered. The active site is altered or changed. Okay, so we can say that when the temperature is higher than its optimum temperature, optimum meaning the temperature at which the enzyme work best at. So when it is above optimum temperature, we can say that the enzyme will denature. Basically, use the correct term. You cannot say that the enzyme will die. You have to say the enzyme will denature. And when it is denature, substrate cannot bind. No more substrate can bind because active site is altered. An enzyme will only work best when the temperature is increased to an optimum temperature, such as 35 degrees Celsius. Okay. Now the second factor that's affecting enzyme activity is pH. Now pH relies on the acidity and the basicity of the blood. So sometimes your blood, if your body cannot maintain a very constant pH throughout the blood, then the enzymes will actually change if they are put in different types of pH. If blood in your body is slightly acidic, then the enzymes will not be able to work anymore. Now this actually depends on whether on different types of enzymes. Some enzymes can work in acid conditions and some enzymes cannot work in acidic conditions. Now it depends on the enzyme that you are trying to explain. So we have to say that acidity and basicity. Now this is called pH. Now pH is basically a measure of hydrogen concentration, hydrogen ions, right? Hydrogen ions. Now this is a little bit of chemical knowledge, but it is basically the measure of hydrogen ions or hydroxide ions. Okay, so we have to know how pH actually affects it. Well, pH affects in the way that temperature affects, but increasing pH or decreasing pH does not really affect the temperature. However, most enzymes work under basic conditions. So basic pH, such as pH. So this could be an pH of around 7, 7.5, 8. Oh yeah, sometimes even 9. It works somewhere around the range of 7 to 8 or 9. Right. Most enzymes, this is most enzymes. Now there are exceptional cases where some enzymes actually work better in acidic conditions. Now acidic conditions meaning if something is acidic, then the pH has to be lower. The pH is lower. The lower the pH, the more acidic. The higher the pH, the more alkaline or the more basic it is. So when pH is lower, there is one enzyme that works. This is one of the proteases that work in your digestive system. Now you will learn this enzyme in the chapter 7 when you learn about human nutrition and digestion in the digestive tract. So this protein is actually called protease. Now that protease name is actually called that protein name is actually called pepsin. Now pepsin is an enzyme that works in acidic conditions, like the acidic conditions of your stomach, right? Stomach has an acidic condition, stomach acid, it works in stomach acid. Now it has a pH of 1, right? It can work in acidic conditions like 0 to 1, 1 to 2, right? So yeah, pH as well, it's the same as temperature. Basically, when the pH is when you use optimum pH, optimum pH, right? It differs from enzyme to enzyme, but if you put the enzyme in its optimum pH, then it will work faster, right? The reaction rate will be higher. Okay, now the next factor that I would like to talk about is enzyme concentration. Now this is very much self-explanatory because the larger the enzymes, the large number of enzymes, the larger the number of enzymes. So you can imagine, like, if there are more enzymes, if there are so many enzymes, how there are many active sites, right? There are many active sites. This means that more substrate molecules can actually bind to the active site of the enzymes. So therefore, this increases the rate of enzyme activity. Right, now this is very self-explanatory. The larger or the increase, if there are many enzymes, then there are many active sites that are waiting to be binded by the substrate molecule. So more substrate molecule can bind to active site. Now the last factor is actually called substrate concentration. So substrates are basically the reactant molecules that will actually go and bind to the enzyme. So if there are lots of substrate, if substrate is a lot or there are more available, then they will bind to the enzyme. This will therefore again increase rate of reaction. Now I should point out for substrate concentration, there's a limiting factor. A limiting factor is basically a factor that is limiting the substrate concentration from reaching highest. So what I want to say is that the limiting factor of substrate concentration is basically the enzyme concentration. Now, let's think about it this way. If there are many substrates but little enzymes, there are not enough active sites for the substrate to bind to, and therefore the rate of reaction will actually not be quicker, but instead it will kind of be slower than if there were less enzyme concentration. Now similarly, it goes the same for enzyme concentration as well. If there are many enzymes, right, but there are little substrate, that means there are free enzymes which are not working. The substrates are not binding to the enzyme. So this means the rate of reaction is actually decreasing, the same as substrate concentration. Now they do like to ask you graph about these enzyme stuff. So let's look at some of the graph. For temperature, remember it works something around 37 degrees Celsius, right? So they could give a graph like this. Now the variable that we're going to change is temperature, and this is the rate of enzyme activity. Now if temperature increases, right, the rate will increases. But however, if it reaches a certain point, such as when in body cells, when it reaches around 37 degrees Celsius, it is working at its maximum temperature, right? It is working at its maximum temperature. However, once you go over its optimum temperature, it's going to start decreasing rapidly, and it's going to go to 0. And when the graph touches 0 at this point, the enzyme has denatured. We can say that the enzyme has denatured. So this is at its optimum temperature. If it works at its optimum temperature, the rate is high. However, if it's over the optimum temperature, it's going to start decreasing rapidly and go to 0. The same goes for pH as well. Now pH actually varies when you draw it out, right? This is the pH, so there could be 1, 2, 3, 4, 5, 6, 7. Now, for the stomach enzyme pepsin, which is a protease, it breaks down protein. The rate will actually be higher at pH 1, and that will go down when it is not pH. Now for substrate concentration and enzyme concentration, it basically follows the same thing. Substrate, let's go for substrate concentration first. So if substrate concentration increases, there are more substrate, meaning it can bind to more, right? So if substrate increase, the rate of reaction will also increase. However, at a certain point, we will not have enough enzymes for the substrate to bind it to. Now at this point, it is the graph flattens out. Well, why is this? They may ask. This is because the enzyme concentration is now the limiting factor. It's limiting this rate, limiting factor. Now the same goes for the enzyme concentration as well. As you increase the enzyme concentration, there are more enzymes, so the enzymes can work faster and more efficiently. However, the graph will also flatten out or level out like this. And at this point, we can say that the substrate concentration is the limiting factor. So these are everything that you have to know about enzymes. So I hope you understand every single thing I try to explain. Once again, thank you for watching these videos. It does take a lot of time to make. So until next time, bye.